TWiM listeners, do you have science you want to share? Microworld.org is the best place to post your microbiology-related news articles, pictures, videos, papers, and more. Sign up for a free account at microworld.org/join and start reaching thousands of microbiology enthusiasts just like yourself.
This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 79, recorded on May 23rd, 2014. Hi, everybody. I'm Vincent Racaniello, and you are listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from Ann Arbor, Michigan, Michele Swanson.
Hello, everybody.
How are you there?
Great. It's going to be a beautiful Memorial Day weekend here in the Midwest.
Oh, yeah. Nice weather?
Beautiful weather.
Yeah, it's going to rain here in the Northeast of the United States. But that's okay. I don't mind. Also joining us today from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
How are you doing down there? Is it Chamber of Commerce weather?
No, it's gone into summer, and we have high humidity, and the afternoon thunderstorms are beginning to boil in, and they're predicting that heavy weather that's been going through the country, you know, the hail and the lightning and the thunderstorms, and we could use the rain, but we don't need the hail or the other associated nasties that go with thunderstorms.
See, here in the Northeast, we don't get hail that much. We don't get tornadoes. We get hurricanes now and then.
Yeah.
But so do you.
It's June 1st is almost here, and that's when hurricane season kicks off, and we pack our hurricane kit, and I'm the designated JLE for my department, so I get to stay behind, make sure all the minus 70s continue to run, and
nice.
keep the liquid nitrogen topped up. And
so all of you must have gone through graduation, right?
Yes.
Because here it's done. Medical school, the undergrad's done. And Ann Arbor, too?
Yeah, the campus has gotten pretty quiet.
Quiet, right? It's kind of nice, isn't it?
It is nice.
Yeah, a little quiet for the summer. Well, all three of us, including Elio, who's not here with us today, still traveling, are just recently returned from Boston, Massachusetts. And there this year was the annual meeting of the American Society for Microbiology, the sponsor of this podcast. What number was it, the 114th?
That's it.
And so I have been going to these for quite a while. I assume you guys have also?
Yep.
This was my 30th meeting. I sat down and counted them up. From the time I was a lowly graduate student in 1982, my first time I presented a paper at a GM, and it was the terrifying 10-minute talk. And it was in those halcyon days when you used 2x2 slides. It was before the invention of posters. If you can imagine, before posters.
What year was that, Michael?
1982.
I got you beat.
Uh-oh.
My first ASM meeting was in Las Vegas in 1978. I have a mug in front of me still. And I'm told this is a collector's item and I should send it to ASM Archives.
Really?
1978. So I was in Peter Palazi's lab. I wanted to, go present something. And back then, the ASM was the meeting to go to for virologists. So I went for a few years. And then, of course, the virologists left.
The schism happened.
Because they wanted half the meeting to themselves, I think. And so they made their own society, the ASV, which is where I went for many years. And then when I got involved with ASM, as a consequence of these podcasts, I started going to ASM meetings again and meeting all of you guys, which I wouldn't have done otherwise.
And now our president-elect is a virologist.
Yeah.
West. Yeah.
And you know the president-elect of ASV? It's me.
Yeah, congratulations.
Thank you. I just think it's cool that, and Lynn and I are members of these societies. We're both presidents. Lynn always says, maybe we can do something. I said, yeah, well, this year, actually, there was quite a bit of good virology at
sure was.
The general meeting. But the sessions I went to on viruses were pretty empty.
Yeah. The rooms were so big, too, so it felt more empty, I think, than it was.
Yeah, the one session I went to, the virology session, was in one of those grand ballrooms, which must seat 2,000 people.
Right.
So, I don't know, were your sessions well attended in general?
So a couple of the ones that I went to were standing room only, overflowing into the next, and they were throwing the slides into the next room via video casting, which is really pretty terrific that the tech has kept up. Not so much with the meeting planners, because the meeting planners can never guess what's going to be hot. So they try to forecast the rooms, and sometimes they get it right and sometimes they get it wrong. But they got the tech all worked out, so you're not deprived of seeing what's actually going on. There were a whole bunch on Ford Doolittle's talk was one of those that was standing room only, and I was sitting in the other room. And
you don't mind not seeing the speaker?
No, I did. I the problem was I was in you have the bargain of figuring out if you're going to sit in one session the whole time or shuttling. And I had the problem. I wanted to see a talk at 8:00. I saw that talk, and then I ran over to see the other talk, and I couldn't get into the room because it was standing room only. So I went. They said, "Well, you can go next door," and so that room was almost full as well. And so I went in there, and it was, you could see the slides, but I didn't see the speaker. And
you know what? In those big rooms, I don't see the speaker anyway, so I just look at the slides. So I don't think that would matter.
And the sound quality, it sounded no different than if he was speaking in the room.
Yeah. So this is a huge meeting, right? There are over 9,000 people, I think.
Yeah.
Do you know, Michele?
I think that the last head count was 8,400.
Yeah, it's a lot of people.
Yep.
And you can't ever see them all, of course. And it's
like a gigantic smorgasbord of science. And you have to just make decisions about what you'll see. But it's a great opportunity to see old colleagues and friends and former students.
Yeah. And as you said, you get a smorgasbord, so you have to pick. But it's one kind of meeting. They're always held in these convention centers, which are in big cities, because they have to be able to accommodate the meetings and all the hotels and all of that. So I enjoy it because I like good science. So I thought we would go through a couple of our favorite talks today and give people a flavor of what went on. I think we did this last year too, right, Michael?
Yeah, we did.
Michele, would you like to start?
I'm happy to start.
You don't really want to, but you're happy to. Okay.
So I'll highlight a session that was Tuesday morning called Bugs as Drugs. And I'll talk about 2 or 3 talks, depending on how much time we have. Starting with one by Dan Portnoy, who's a professor at Berkeley. So Dan is really been at the forefront of a field that we now refer to as cellular microbiology, which is trying to understand the cell biology related to infections. And he trained with Stanley Falco, who has a great quote that really motivated many of us to join this field. And that is that the examination of the cell biology of microbial invasion reveals not so much a belligerent event, but rather a view that many of the bacteria we associate with disease are actually quite remarkable cell biologists who possess admirable insight into the fine points of eukaryotic life. So that's Stanley Falco's wisdom. And I think Dan is just a great example of somebody who's illustrated that with his research. So his lab has been studying Listeria monocytogenes, which some of our listeners may recognize as the cause of a foodborne illness that is especially risky for immunocompromised and also pregnant women, because this microbe, once it gets into the stomach, can actually invade deeper tissues. And for example, it can cross the placenta and infect the fetus and result in stillborn. So it's a serious problem. It's also tricky because the bacterium can tolerate heat, and it can also continue to grow even in foods that are refrigerated. So it's a real challenge for the food industry. But by studying its pathogenesis, Dan and a number of colleagues in the field have been able to exploit this pathogen as a a tool for immunotherapy and for vaccines. So I'll describe some of the key features that allowed them to do that.
Before you go on, Michele, just let me interject. We did a TWiM number 4 on cantaloupes and listeria. So if any of our listeners want to go back and listen to cantaloupes and listeria, they can check out some of the foodborne issues associated with listeria.
So 2 of the factors that allow this bacterium to cause invasive disease one is called internalin, and this is a protein that allows listeria to bind tightly to eukaryotic cells and invade them. So this is a tool that it uses to get into deeper tissues. It also has a pore-forming toxin that allows it to lyse from the vacuole that it is ingested within, a phagosome, and in so doing, it can escape into the cytoplasm, the cytosol of the cell, and therefore escape being delivered to the lysosomes, which is the stomach of a cell and can degrade many microbes. Once listeria is in the cytoplasm, it has a protein called ACT-A that expresses just on its tail end, and it recruits short filaments of host actin to that region, which in effect equip the bacteria to be propelled throughout the cell and push out of one cell and into a neighboring cell. So it can actually propel itself into a neighboring cell without being exposed to antibody, neutrophils, antibiotics that are detrimental to bacteria. So this bug is very good at getting into cells, and then once it's inside, gets into the cytoplasm and can spread. What Dan's lab and others have realized is that because this bug can get into the cytoplasm, it is perfectly situated to deliver antigen to the cell-mediated immune pathway, which requires that antigens be presented to the MHC1 pathway in the cytosol. So what they've done is engineered a mutant of listeria that no longer has the internalin A protein, so it can't invade non-specifically into other cells, and it doesn't have ACT-A, so it can't spread cell to cell. So this mutant now can only be taken up, or is most efficiently taken up, by antigen-presenting cells, macrophages, dendritic cells. It can still pop out of its vacuole and get into the cytoplasm, so they've engineered this now to secrete antigens of choice into the cytosol, which are then presented and can recruit a specific class of white blood cells called T cells that can specifically recognize and kill either infected cells or tumor cells. So the project that he focused on in his talk is a project that allows them to use listeria as immunotherapy against particular cancers. And he described that by first mutating this internalin A and ACT-A of listeria, they then did safety tests in trials in humans to make sure that the bacterium could be tolerated and would not cause disease. And remarkably, they found that you can inject up to 10 to the 9th of these mutant listeria into the bloodstream and not cause disease or any symptoms. So having established its safety, they then engineered a strain to express an antigen that is on the surface of particular cancer cells but not on normal cells. And in particular, they focused on antigens that distinguish pancreatic cancer cells from normal cells. And in human trials, they were able to find in a very small cohort of patients that the delivery of this engineered strain of listeria conferred protection. I should say it prolonged the life of this cohort of patients that had severe cancers, pancreatic cancer, mesothelioma cancers. So they are now have advanced to phase 2 trials where they're going to increase the cohort of patients and also adjust the protocol to directly test the capacity of this listeria vector to deliver antigen to the cytoplasm, trigger cell-mediated immunity that will then stimulate T cells to seek out and kill cancer cells in people. So what's cool about this is it really exploits some really beautiful genetics and cell biology that scientists in academia have done for the last decade. And in addition, a number of people who've trained in Dan's lab as PhD students have now gone on to form companies that are taking this knowledge to the next step. So, for example, Peter Lauer is at Aduro Biotech, along with Justin Scoble and Chris Ray, three of Dan's former trainees, and they're the CSO of Aduro Biotech, Tom Dubinsky, now closely collaborates with Dan, and they're in turn working with a group at Johns Hopkins to organize the clinical trials. So this was a really beautiful example of basic science, understanding the mechanisms of pathogenesis, taking it to biotech, and then on to clinical trials. And the early signs look really promising for treatment of pancreatic and other cancers.
Wow, that's cool. I didn't know that. Many people are using viruses to do similar things to target, to kill tumors directly. But
right.
So, do you know offhand what is the surface antigen that distinguishes these tumors?
Well, the one he talked about in his seminar was mesothelioma is the disease, and so it's mesotheliin, I believe, was the protein. That's
pretty cool.
Yeah.
And so the bacteria on their own are not at all pathogenic in people, right?
That's right. Well, the wild type bacterium is, but once they mutate, the ACT-A that drives the cell-to-cell spread, and then the internalin A, which allows it to get into non-phagocytic cells, then it's quite safe.
Do they inject it directly into the tumor or IV, or how was it given, do you know?
They, in the safety trials, they gave it IV, and it was found to be safe. And I don't recall, I believe that's what they're doing in these trials as well, although I'd have to look at the paper to double check.
Nice.
Certainly this bacterium can survive in macrophages, so you could put it in the bloodstream and it could be carried to other deeper tissues that way.
Yeah, neat.
If you're out there looking for visual of what Listeria is doing, just using your keyword phrases, actin rockets.
Yep.
And cool. You can see what the Listeria are actually doing once they get inside our cells. Some of the movies that are out on YouTube are absolutely fascinating, and they really bring what Dan Portnoy and his colleagues have really discovered, and it really drives home Falco's quote about how clever these little creatures are.
So it's Julie Theriot. I can direct people. I use these in my med school lectures all the time. Her name is spelled Theriot. She's at Stanford. She's a Howard Hughes investigator, and she also won a MacArthur Genius Award. And she is a kind of a biophysicist who's been studying been exploiting Listeria to study kind of biophysical properties of actin, and she's posted a number of really beautiful movies on her website.
I have two brief stories on Dan Portnoy. Once I was at a meeting in Switzerland with him, and I remember he starts his talk by Listeria, it won't kill you, but you wish it had. And then I roomed with him at that meeting, and what I remember is he just talked all night.
Yep. He's got a lot of energy.
So we were in the same room, and I was trying to go to sleep, lights out, and he would say something, and then he would stop, and then I'm falling asleep, and then all of a sudden he starts saying something else.
Yeah. He has also, I think, in the microbial pathogenesis field, he may have placed more trainees in academic faculty positions all over the country than really anyone else. He's just an excellent mentor and such a creative thinker. It's been a real boon to the field.
Neat. All right. Hey, you want to do a round robin? Do one each? How about that?
Sure.
Yeah.
Michael.
Well, I'm going to start out with a story that made me infamous on my birthday. I actually
Happy birthday.
It was Tuesday.
Yeah, well, late better than never, right?
Better than never. And so,
how old are you? Are you 52?
No. I'm approaching the 29th anniversary of my 29th birthday. One more year and I'll be 58. But I'm 57 now. So someone pointed out to me, one of my colleagues at work is a year older than me, and she said, I'm celebrating the 29th year of my 29th birthday.
It's cool.
And she said, next year you'll get to experience this. So I laughed. And so many of you probably saw this on the news. It was the big scare, so to speak, of the assessment of survival and transmission of selected pathogens on aircraft cabin surfaces. And this was a really neat poster produced by K.A. Valaganoff and I.H. Chen, A.N. Goodlow, B. Konsei, P.J. Zwack, V. Cheng, J. Chu, M. Henderson, R.A. Overfelt, and Jim Barbary, all of the Auburn University micro department. And what they did is they're very much concerned about the built environment being able to transmit infectious agents. And they specifically looked at two: MRSA, methicillin-resistant Staph aureus, which many of us know is an antibiotic resistant microbe that causes all sorts of bad things in the hospital. And the other one is the famous jack-in-the-box organism, E. coli 0157H7, the one that we're hearing about recalls in hamburger this weekend because there's another 157 outbreak that everyone's concerned about with ground beef. And so what they did is they got some common aircraft surfaces from the airlines, and they actually deposited a concentration of 10 to the 5 cells of these two specific pathogens onto these coupons. And they used a rubber armrest, some metal toilet handle, a plastic tray, a plastic window shade, the leather seat, and the seat back cloth. And they simply asked a question: how long will these microbes survive on these common surfaces on a plane? So this was all in vitro work, had nothing to do with airplanes. So this was done in their lab. And they stressed over and over in their ASM Live session that this was intended to establish a baseline in order to set a risk of what the FAA and airlines should be looking for. And what they learned is that MRSA survives significantly longer on pocket cloth. And if you think about it, you know, the aircraft fabrics are principally polyester, and you've probably heard me say that the only thing Staph aureus likes better than people is plastic, and polyester is plastic. You need only talk to one of our service people this weekend and ask them whether or not their polyester uniforms are breathing properly. I always hear from my Navy friends around Charleston about the polyester cloth and how it's so hot. And E. coli 0157H7 lasted on these surfaces on the armrest, which is a plastic that's not quite smooth, but it's that little bumpy plastic so you can grip it. And it lasted about 96 hours. And so their principal conclusion was that their study provided insight into the relative risk of infection from these contaminated surfaces. And so what the reporter, David Curley from ABC News, was asking me about was, am I concerned about flying? And my answer was no, simply because I followed the advice my mother gave me when I was a toddler. And after I use the restroom, I wash my hands, and before I eat anything, I wash my hands. So the sage advice I gave, I don't know if it made it through the interview or not, but the advice I gave was what I typically do when I'm flying in order to be safe is I wash my hands after using the restroom, exit the restroom, and then I keep a little hand sanitizer with me in my pocket. And I apply that after I have refastened my seatbelt and lowered the tray table before I eat. And then I apply the hand sanitizer without touching those objects because I know how sticky the microbes are, and I want to prevent fecal-oral transmission of the E. coli. And I also want to not pick up any of the nasty bacteria that can be transmitted by these fomites. And so that was the big news.
So, Michael, do you also avoid licking the upholstery on your airline seat?
I do that. I do, but I have flown enough to see enough people actually, lick their fingers after eating salty pretzels or something to clean them off because they can't get up because of turbulence. But this paper or poster was just one of many that are beginning to explore the built environment. The other one that I think the news media should have picked up on was one by Cami Wagner of UNC Wilmington. And she is an undergraduate, so this was undergraduate-based research. She's at UNC Wilmington in Wilmington, North Carolina. And the title of her poster was The Isolation and Characterization of Staph Species on Surfaces in a University Weightlifting Facility. And so Cami, who happens to be one of the mentees of my former graduate student, who I didn't know when I went to see the poster, because the way the search engine for the ASM app worked is you only saw the first offer and you didn't see all the other offers. And so I went to see this poster because I was interested in this topic. And Cami actually went to the university weight room and she collected samples and asked the real question of whether or not the weight room does indeed have MRSA and other scary staph. And so she went to a weightlifting center. The surfaces were subdivided into smooth and rough textures, very similar to what we saw at Auburn. And she then plated them on traditional media like mannitol salts agar. And she also then went the next step and went, because she was out in the wild collecting wild animals, if you will, from the weightlifting facility. And she used 16S, 23S intergenic regions to look to see who was there from the staphylococcal world. And so she did a really good poster going from basic bacteriology, looking on normal Petri plates, to doing fundamental molecular biology. And her conclusions were, based on the most frequently contaminated fomites, it is possible that texture surfaces allowed better adherence and or were more difficult to clean, which makes perfect sense. Those nooks and crannies are difficult to get cleaned out. And what she learned is she found several species of Staph aureus, Staph epi, Saprophyticus, Staph capitis, which is normally associated with your head, and Warneri were isolated from these various surfaces. So this new up-and-coming scientist did this really neat paper, followed all the strict guidelines, and I'm going to contrast her work against the gold standard, which is a paper by Angie Collette from the CDC. So I hold the CDC as my gold standard in sampling. And you may not think sampling is cutting edge, but the science that was at the general meeting is really cutting edge. And the CDC put out a cutting edge paper about how to do sampling, and where they plant a defined concentration. And what they were looking at is Acinetobacter baumannii and MRSA. And they were asking how many, if you put a defined concentration on a surface, can you recover? And they did some pretty sophisticated experiments where they looked at the volume of the bacteria in the sample that they deposited, and they looked at the recovery rates. So they were using, again, concentrations that you typically see in the environment, about 10,000 colony-forming units per milliliter. So this is a viable cell count. And then they used sponge wipes to see how many they could recover, and they plated them on various geometries going from, and I got to check my notes here so I don't screw it up. Coupon sizes were 50, 100, 200, and 350 square inches. And so as you would spread the concentration out, you looked at recovery rates. And so they actually published the gold standard of how to do some of the studies that Jim Barbary and his colleagues at Auburn were doing, and how Cami Wagner and her future colleagues are going to be doing in things like weight rooms, airplanes, And where I commonly sample is hospitals. So collectively, these 3 posters did a real service for looking at the built environment of determining whether or not it does indeed represent a risk and what we can do to protect ourselves.
I think last year or maybe the year before there was one on hotel rooms, right?
Yeah, the TV remote.
The remote is the most contaminated thing in a hotel room, right?
Right, because it lives in the bed with you. Most people in hotel rooms...
Not mine.
Not mine.
Well, in hotel rooms, you sit on the bed and you change the channels because you can never figure out which station is which.
I lay in bed and listen to TWiM and TWiV.
Oh,
This is... where should I send the check, Michele?
a shameless plug, but
I don't
you'll get a
watch...
check
I never
from Ray.
touch... I never touch the remote in the hotel. I have no desire. There's plenty of other things to do, but I know most people do. Yeah.
Yeah. So these 3 papers the 3 posters were just phenomenal. One, if you've never been to one of these large meetings, the only thing I can say is pack your roller skates because there's aisles and aisles of posters and there's so much science and so little time.
I think we know that these common areas in hotels, airplanes, trains, offices, wherever there are a lot of people, subways, they're going to be contaminated. And just by virtue of the fact that there's always a constant flow of people, they're going to refresh whatever populations are there.
Right.
So this is not surprising. The question is, what do you do? I guess what you said before, you just have good hand hygiene, right?
Good hand hygiene, and actually one of the things that the NFL, the National Football League, has learned about weight rooms and the issues associated with protecting their prized athletes is they learned something simple. You just bleach the towels to prevent the spread of these nasty pathogens.
Well, the floors are a mess. People get athlete's foot all the time. I guess everyone should play golf. There's no problem there, right?
Unless you rent the shoes.
Oh my gosh.
That's bowling. I'm sorry, that's bowling.
Yeah, that's right. Bowling shoes. I'm not going bowling.
Let's not forget,
anymore.
we have a pretty good immune system.
Yeah.
Your skin is a great barrier.
You're right.
You don't want to touch the subway turnstile and then touch your eyes or your nose, though.
Exactly. Wash your hands.
Give the bugs access.
That's it. Just have some common sense. If you have cuts on your hands or skin, be careful. Common sense. All right. Let's see. What should I do? I have, I went to the opening session on Sunday or Saturday. Sat next to you, Michael.
Yeah.
And I wanted to talk about a talk by Ove Hoag Goldberg, who's from Australia. And his talk was Climate Change and Unraveling of Microbial Partnerships in the Ocean. And he said several times that his talk was going to be very depressing and that he was very negative. And this is all about what climate change is doing to coral reefs, mainly, is what he talked about. And he said, I think at the very beginning, there should be no further debate about the reality of climate change. There's no question about it. And anyone who doesn't think it's happening is simply wrong. And he focused on the oceans, and he talked about how greenhouse gas levels, they're rising as a consequence of human activity, burning fossil fuels, etc., have been increasing global average temperatures by 0.2 degrees Celsius for each decade over the past 30 years. And most of this excess heat is absorbed by the oceans. It's the top 700 meters of the ocean. Its temperature has gone up 0.6 degrees. And he said the amount of energy being absorbed by the ocean every second is equal to 4 Hiroshima-sized atomic bombs.
That was so impressive.
Wow. It was amazing.
What a great illustration.
So, and he said basically for years they did absorb a lot of heat, but now they're becoming... It's rising the overall temperature. They're also absorbing a third of the carbon dioxide produced by human activities. That causes the pH to drop, and it's dropped 0.1 pH units since the pre-industrial period, but he said that is a large amount because it causes a substantial decline in carbonate ion concentrations, which has huge biological effects. And one of the things he said that impressed me is that we're pretty sure that these kind of changes have not happened for millions of years. The Earth has
since the dinosaurs.
Yeah, the Earth has undergone various periods of climate change and so forth. But he said things on this magnitude haven't happened for a long time, right? And he also mentioned other sorts of changes like sea level rise, polar ice melts, etc. And what he focused on is the effects of these changes on corals, which support a lot of ocean life. And corals, of course, have an organism in them, and there's a symbiosis between those organisms and algal protists, which live inside them. They're called zooxanthellae. And so this is a symbiotic relationship. When the temperature rises and the carbonate concentrations drop, these symbionts leave, and they're brown. So the coral turns white. They call it coral bleaching, and the coral dies, basically. So he showed pictures of acres and acres of bleached coral. And so no more coral, all the ocean life goes away. And he showed a series of photos of what happens. You have this beautiful coral reef with fish and all kinds of life around it. And then it starts to become green as the coral leave and algae come in. And I think he said, this is great for those of you who want to study algae. But then the last phase, everything is dead. And he said in 50 years, at the current pace, there's going to be no more coral left on the globe. So I just think this is really a very sad state of affairs, and I'm not sure that we can do anything about it, because burning fossil fuels is what many countries have to do to become developed. So,
and it's, I was talking to one of my colleagues in the exhibit floor, and he was pointing out to me that the fossil fuels waste most of their heat, waste most of their energy. It's not a very efficient way of harvesting the energy from the consumption. And if you look at the heat delta, he pointed out there's the DOE has on their energy site how much coal and fossil fuels are being used, and the number that is lost to heat is the equivalent of using the fossil fuels and the coal. Fossil fuels being oil and gas plus the coal. We're losing 50% of our energy due in the conversion process to heat, and that's how much coal, oil, and gas is used in our energy equation. So if we could just figure out efficiencies, so when you are complaining about these new fluorescent bulbs or LED emitting lights, any increase in efficiency could, change our dependence upon these fossil fuels. And that's what we really need to do, because it's the excess CO2 that's really driving this system.
Right.
And harness microbes to solve this problem.
Oh, that was one of the coolest quotes I picked up in, I went to the session on metals, and I got the number I've been hunting for. The number of electrons flowing through a bacterial membrane per second are on the order of 10 to the 6th to 10 to the 7th electrons are flowing through a membrane per second. And I've been hunting for that number, and there I go to a session, and out it pops from Ken Nielsen's mouth. And I couldn't have been more delighted, because it helps with my current story on what we're looking at. So it really shows how efficient microbes really are at making use of what they were given, and sunlight and carbon, and they just do what they need to do. But they've had more time to figure it out than we have. They've had billions of years.
Michele, back to you.
Yeah, I'm going to turn to something a little upbeat after that depressing story. So Rob Britton, who is a professor at Michigan State University, chaired the Bugs as Drugs session, and he described his work on Lactobacillus reuteri and promising data showing that in a mouse model it can suppress bone loss. So the syndrome that they're interested in treating is osteoporosis. So women, the statistics are that half of women over the age of 50 will suffer from osteoporosis and be at risk for bone fractures. And I'll bet that number goes up as the baby boom generation who's more active will have estrogen-related bone loss, but retain their love of skiing and running and cycling and all kinds of other active sports. Anyway, so Lactobacillus reuteri is a common microbe in foods, and they have found that it has an impact on two different cell types in our bones that dictate whether we're growing bone or resorbing bone, losing bone mass. So the two cell types are called osteoclasts. They're the ones that absorb bone, and osteoblasts, which are the cells that make bone. And they found that Lactobacillus reuteri releases a factor that, by some mechanism, it has an impact on CD4 T cells that is increasing the osteoblasts and decreasing the osteoclasts. So, as a practical matter, they find that mice that are fed Lactobacillus reuteri have a change in their microbial community in their gut, and they also have less inflammatory markers, and they have less resorption by the osteoclasts, and therefore increased bone mass. So the hope is that, in addition to, we've heard about calcium and vitamin D as being important for bone health. The hope is that we can identify the factors made by Lactobacillus reuteri and be able to influence the balance between resorption and bone formation with this probiotic, and therefore treat through a simple dietary supplement, this osteoporosis, which can affect so many otherwise healthy women.
That's neat. And this is in clinical trial, or?
I don't know if it's in clinical trial. The data that he described, and some of it's published now, is in a mouse model. So they're learning the mechanism there, and then we'll use that information to design specific tests in humans.
Nice. Michael.
Well, since it's nearing dinner time here on the East Coast, I thought I'd talk about the farm to table session. The one that I found absolutely fascinating in terms of presenting the current problem was Peter Fang, who was from the FDA, and he was talking about the whole issue of how safe our food really is, and how our lifestyles are actually responsible for some of the changes that we're seeing with outbreaks. Principally, he introduced this concept to me that I sort of thought about, but I hadn't really understood the magnitude of the ready-to-eat vegetable market. We've probably all had bagged lettuce, bagged spinach, bagged Caesar salad, and I had no idea how much the United States and the developed world was using in terms of these bagged salads. We're using close to 3 million bags of this stuff per day. And oftentimes this fresh produce has been harvested and washed in the field, and it's truly going from the farm to your table in a record period of time. And as we have developed advances in packaging, so has the microbe come from the farm to the table, because it's literally surviving on these small pieces of soil and detritus that are associated with this vegetable matter. And he went through some of the scary things that are happening, and he's specifically talking about the variations of the E. coli, and he introduced the concept of what's going on with the sprouts. And I asked him a question during the session about sprouts, and he said, well, oftentimes sprouts, they didn't wash the seeds, and the seeds are very challenging to clean, and the seeds are germinated, and oftentimes they weren't monitoring the water that was used for the germination. But that's all been addressed now. And so the sprouts haven't been in the news as much. And he went into acidic foods like cider and goat cheese and salami beginning to cause problems, and how the microbes are adapting. It was just a fascinating story of what's going on. And the take-home messages are our food supply is wonderfully safe, considering how much of this ready-to-eat food product is present. And he ended by forecasting the next thing that may actually make it into, the foodborne illness category. And he talked about smoothies at the end. More and more of us are taking that quick drink in the morning where we're putting in vegetable matter to whiz it up with bananas and orange juice, and we're throwing in some lettuce and some spinach and whizzing everything up. And he said smoothies may be the next big thing. And that's how he ended it.
Because we're not cooking the spinach or the kale. Mm-hmm.
That's right. It's the raw foods that we're beginning to introduce. But at the end of the day, we're relatively safe. And the speaker that followed him was Roy Curtis, the great geneticist that I always associated coming out of WashU, and now he's at Arizona State. And I didn't know half of the things that Roy shared with us about his life. He gave a life history of how the microbes and the animals have evolved literally under his guidance.
That was great. I like that. I went to that session to hear him, and I caught the previous one that you just described. And I have to tell you, whenever I get these bags of lettuce, I always look at it and they say triple washed, and I go, ha. I always wash them. And my wife, who is a scientist, was trained as a microbiologist, she always says, you don't have to wash that. And I say, what, are you kidding?
My gosh.
Now I have
evidence.
I have ammunition. I'm going to tell her I went to this talk and I learned that you have to wash. I mean, he showed a picture of a leaf that came out of one of these bags that was all covered in brown stuff.
And you just eat it. And the other thing people should appreciate is that the restaurant industry, because labor is the principal cost associated with restaurants, they're using a lot of these bagged products. It's not, one serving size for us. It's literally for the entire night for the restaurant.
Yeah.
And these breathable plastics, it's truly been the advances of packaging that have made all of this possible, has helped keep the cost of restaurant-prepared food down because they just open it and they literally add the dressings.
Yep.
And so, America, be forewarned, but our food is safe.
Convenience. All right, I will highlight another talk from that opening session on Saturday by Laura Hooper, and her talk was "Good Fences Make Good Neighbors." And she is from Southwestern Medical Center in Dallas. She's a colleague of Julie Pfeiffer, who also gave another talk in another session, who also was my guest on our TWiM from ASMGM, which is coming up this weekend.
She earned her PhD in my department here at Michigan.
Yeah, that's right, with Alice. Yeah, I asked her to tell her history, and she told us that, right.
And Laura came out of the Gordon Lab out of WashU.
Mm-hmm, mm-hmm. Yes, it's only 5 or 6 degrees of separation in science.
That's right.
So Laura and Julie collaborate because Laura has a mouse colony, a germ-free mouse colony there in Texas, in Dallas, and they use it for her work and for Julie's as well. And Laura is interested in why we can have all these bacteria in our intestines and we're okay. So what limits the ability of these bacteria to cross the intestinal barrier and to get into host tissues? And she had talked about a family of secreted C-type lectins. These are lectins, proteins that bind carbohydrates, and the C-types require calcium. And she talked about a family called Reg3, which bind peptidoglycan, surface peptidoglycan from bacteria, and this binding kills the bacteria. So when bacteria get too close to the epithelia, when they start to invade the cells in the intestine, this triggers production of this lectin, and that tells the bacteria to back off because it kills them. And she talked about the mechanism of killing. Basically, one of the Reg3 lectins called Reg3 alpha, it binds phospholipids in the membrane of the bacteria and forms a hexameric pore that permeabilizes the membrane. And Michael was sitting next to me. And what did you say? It destroys the electron potential?
Right. Collapses it.
Collapses the electron potential. You were exclaiming all the time. You like that talk.
Oh, that talk was just off the hook as far as I was concerned. This Reg3 gamma family of proteins is islet regenerating factor, and so you begin to see the wonder that this, and she displayed the wonder of her science in all its glory at that opening session, and I cannot imagine talking to 10,000 people because the room was packed.
It was. So Reg3 alpha is inhibited by a lipopolysaccharide, and she thinks that's why Reg3 alpha is bactericidal for gram-positive but not gram-negative bacteria. She also showed these cool pictures of the mucosa of the intestine. There's about a 50 micron gap between the epithelial cells and the bacteria. She called it the DMZ, the Demilitarized Zone, and it's maintained by the cells sensing the bacteria. And in fact, in mice lacking a sensor called a protein called MyD88, this zone is gone. MyD88 is an adapter protein that's used by most toll-like receptors, which sense bacterial products and initiate production of cytokines. In mice without MyD88, there's no innate response to bacteria, so this 50 micron gap collapses. I thought that was really cool.
And if you think about it, the average rod is about a half a micron across. You know, we always tell people at flu time to keep everybody at arm's length so that you can avoid getting the flu. But what our intestine is doing is keeping these evil gram-positives 100 cells away from us, 100 body lengths away, really setting up her metaphor of a demilitarized zone, I think, is spot on, because 100 cells is a tremendous gap if the average width is only a half a micron.
So a lot of this work was done in germ-free mice, and Julie Pfeiffer has used these same animals to show that you need the bacteria in the gut to get efficient polio infection. And she talked about that in her talk, and we discussed it on TWiM 286, which will be up this coming Sunday. All right, you want to do one more round?
Sure.
I do. Actually, mine will be short because it follows directly off the Demilitarized Zone concept. This was also in the Bugs as Drugs, and it was a talk by Patrice Cane from Brussels. And he and his group have been interested in the impact of the microbiome in the gut on obesity, among other things. But just a bit of hopeful news. They've identified a particular species that in mice not only correlates with reduction in obesity, but also if you first take an obese mouse and then feed it this species, I can't say it very well, Akkermansia mucinophilia, they find that that's having that species in your diet can reverse a number of markers of ill health, including in obese mice, they had reduced fat mass, they had reduced inflammation in their adipose tissue, reduced insulin resistance, and reduced plasma cholesterol. And they're beginning to understand the mechanism, but it correlates with increased mucus production by the goblet cells in the gut. So an increase in this barrier or demilitarized zone that you spoke of. And there is some connection to production of antimicrobial peptides, including molecules from the Reg3G family. So there's hope that we'll be able to use this as a probiotic to actually reverse some of the damage and inflammation that is associated with obesity in humans.
Cool.
But again, this is all in a mouse model.
And that's one of the underlying themes that was going through a lot of the microbiome discussions in the poster sessions. And there was a tremendous number of talks on the human microbiome, and everything is everyone's trying to come up with that magic pill. So there was even a session on poop pills, I think. I didn't quite make it there, but everyone is looking at how to make these probiotics so that they can move away from stool transplants to actually making it more into what we anticipate as ethical pharmaceuticals.
Yeah, and I haven't been to a lot of these talks, but this is the first one that I attended where they introduced just a single species and were able to reverse many of the markers of obesity. Again, in a mouse model, but nevertheless, it's hopeful. I think we're making progress.
It's neat. Very cool. Michael.
Well, the last one I want to talk about is the genetic characterization of strep pneumo desiccation tolerance. This was another poster board that I went off to, and we all know that strep pneumo is a common colonizer of our nasopharynx, and it spreads person to person. This is big news for community-acquired pneumonia amongst the elderly and hospital-acquired pneumonia amongst those that are hospitalized. And what these folks did is they first asked the simple question, how long can strep pneumo survive desiccation? And they learned it's about 28 days, again, making the old argument that cleaning is good. But then they looked at the genetics. And so here's where I got really excited. They did old-fashioned insertional transposon-based mutagenesis to make a library of strep pneumo using serotype 2 strain D39. And then they asked the question, they desiccated it for 4 days, and then they used the TriSeq, the transposon seeking system, to identify several classes of genes. And now here's the pop quiz. What genes do you think their screen uncovered to prevent death from desiccation? And it's the ones you'd normally guess. It's the ones that protect the cell from UV damage, damage associated with hydrogen peroxide, and then, of course, the genes that protect cells from osmotic shock. So as you lose water, you would turn on those repair systems to take care of it. And as you're sitting out in the environment, ultraviolet light, of course, is a big thing. And so their principal conclusion is they confirmed that nucleotide excision and repair pathway facilitates the survival of these microbes to desiccation, whereas pyruvate oxidase reduces pneumococcal survival. So this was, a first of its kind study demonstrating the importance in strep pneumo, which is a big pathogen, that desiccation tolerance is real. I mean, these microbes do indeed have the ability to survive in the environment for a long period of time, and DNA repair and stress response pathways are essential for these organisms to stick around in the environment and be the bad actors that they have become. And so the things that you learn theoretically about transposon-based mutagenesis and, how it can fit, you just wandering the poster areas, my keyword searches didn't pick these up. It's as I was just going down, scanning the titles, I stumbled into these, and that's I think posters should be a little longer in time. They were only an hour and a half, and I could have used 3 hours in each of the poster sessions just to chat up the authors. And that's one of the neat things I like to do, is to talk to people. And a lot of them didn't see my name, but they recognized my voice, which was very peculiar. And we had groupies out there, Vincent.
That's right.
Groupies.
That's right. Just recognize your voice. My last one here is a talk by Jesse Bloom on molecular constraints on evolution of influenza virus. So the idea here is, what constrains evolution? It's thought that as evolution occurs by selection of mutations that arise during error-prone nucleic acid replication. The idea has always been that you have to have certain mutations to get other ones later. So there's this continuum. There's this famous analogy by John Maynard Smith where he compared protein evolution to a word game where you start with a word, a 4-letter word, and you change a letter to make it a different word, but it has to be a real word. So you can go from word to war to gore to gone to gene. But if you put in changes that end up in the last word early, they wouldn't work because they wouldn't make words. So the idea is it's the same thing in evolution. You have to have certain mutations before others. So they test this in influenza virus using one of the genes that encodes the nucleoprotein. And from 1968 to 2007 in the H3N2 virus, there were 39 amino acid changes in this protein. Since we have sequences every year from influenza viruses, they could reconstruct the sequence of changes as they occur from year to year, about a couple each year. So they basically made 39 viruses which recapitulate this sequence changes from year to year to year, with one mutation, 2, 3, 4, 5, etc. And then since this NP protein is involved in a viral RNA synthesis, they did an RNA synthesis assay. And what they find is that all 39 NP proteins are just absolutely fine at making RNA, just like the parent strain from 1968. Okay. Now they introduced each change singly into the NP protein. All but 3 were fine in terms of RNA synthesis. 3 of them really messed up RNA synthesis. And those 3 amino acid changes made the protein less thermostable, and the protein levels in cells were much lower. All right. When they put those changes into viruses, it reduced viral replication. Now, this is an artificial situation because you're putting these mutations in individually. If they took each of those 3 bad mutations and then put in the changes that had occurred before that in time, the protein was fine and the viruses containing them was fine. So in other words, these 3 mutations were selected at some point for some selective reason. They actually think they're selected to escape cytotoxic T-cell recognition. The only way they could arise was if previous compensatory mutations arose.
Right.
So you have to have a sequence of events, and evolution is not totally random. Right, so you have some changes occurring that facilitate the emergence of others. So this is it's really one of the first ways where this has been looked at comprehensively and gives really nice evidence that epistasis, which is the interaction between different mutations, at least in this case, is based on structural stability of a protein. Very cool stuff.
It makes sense that it would work that way. Now, can you go the next level and run the risk assessment on this and ask the question, what is the likelihood that one of these, for example, there was a lot of buzz around the Middle Eastern respiratory syndrome virus and whether or not right now it doesn't transmit well to people, but could you take the sequences that are associated with the proteins that facilitate its infectivity in people and run them through this model to say, will this bug virus / change to become more easily transmitted?
Well, we don't know what regulates the transmission of
I know.
These viruses. That's the problem, right?
That's
We don't even know for influenza what regulates transmission in people. So that's why it's really hard to make predictions about when. That's why Ian Lipkin at ASM Live, when asked by Stan Malloy, is MERS corona the next pandemic? He said, I don't have a crystal ball.
Well, that's really important because there is so much we don't know, and our blessed funding agencies are actually asking us to do more translational based research, but there's still a lot of fundamental research that must be done.
Absolutely. And that gives you the answers to the translational stuff. I don't see why we have to cut back on the basic stuff.
Yeah, as illustrated so beautifully by Dan Portnoy's work on listeria.
That's right. That's right.
Listen, before we go out, could I mention a session that I attended at the American Society for Microbiology Conference for Undergraduate Educators that Vincent and I both attended? I was delighted to see there was a microbrew session, so these are their short sessions, by Dr. Stacy Latina at Gwyneth Mercy University in Gwyneth Valley, PA, entitled TWiM, TWIP, and TWiV.
Wow.
How I Use Podcasts to Excite Students About Current Microbiological Research.
Cool.
So she gave a session and had a handout that basically described how she has used our podcast to teach undergraduates how to read the primary literature and become skilled at evaluating it, being able to pick out the main points. And what was neat is she described after they take her training in her course, she then has talked to other colleagues who teach more advanced courses who report that students who have been in her course and have been through this podcast-based literature reading now do much better on any exercises where they have to read primary literature and review the papers.
Nice. I love it.
It's delightful to see that creative professors like Dr. Stacy Latini are using our podcasts in really creative ways to help students.
Love to get that handout. I'm going to email her.
I will, or I can fax it to you.
That'd be great. All right, let me just read a couple of emails here. First one is from Matthew, who writes, Dear Doctors, after listening to the second portion of TWiM 78, talking about the presence of gram-negative nosocomials around Brooklyn, I noticed several people wearing scrubs while at lunch near a hospital in Houston, Texas. Then a thought occurred to me, disease everywhere. Might be part of the problem if these microbes are moving out from the hospital, be that they're carried from a day's worn clothes. I realize I'm asking to speculate, but think this might be a good consideration for hospital policy. Houston is currently 27 centigrade, 50% humidity. P.S. I finished my sandwich. It was a pastrami Reuben.
Ooh, sounds good.
We talked about this before, about how I used to see people walking around here with scrubs, but it seems to be less recently. Then someone sent us an email saying they saw people on buses, and they actually sent us a picture. Michael, could that be a way?
That is a way. And in fact, our colleagues at APEC, the Association for Professionals in Infection Control, which is a society like ASM with 30,000 members, is very much concerned about clothing in the hospital. And we've all seen our colleagues wear their little hats into the cafeteria and their evil shoe covers. Yep. Because the floor, we all know, is not clean. And the reason you're wearing shoe covers is for the obvious reason. And so I think, Matt actually got the point of the paper is, it's the inverse square law. The closer you are to the hospital, the greater at risk you are of picking something up from the hospital.
Well, the hospital should make a regulation to not allow people to leave with scrubs on, right? Yeah.
That's the, it's the unintended consequence. I don't think people appreciate that they're doing this, but it's an unintended consequence. And these multi-drug resistant creatures are doing it. And those of you skilled in math, run the model and ask, if we prevent the microbes from leaving the hospital, how long will we get to use antibiotics? Another 10 years? Another 20 years?
Next one is from Jim, who writes, Hi, folks. Ideas with Paul Kennedy on the Canadian Broadcast Corp site recently ran a 54-minute program about invasive species that included a short reference to rock snot. At the link, find the title Bioinvasion, Attack of the Alien Species to download it. I thought it, along with Professor Schmidt's comment about furry teeth, are a good intro to any program for kids about biofilms.
Absolutely.
Yep. And finally, Peter writes, you have spoken before on TWiM about the potential risks of triclosan-resistant pathogens developing through its overuse. A recent open access article from the University of Michigan.
Go Blue.
You're so funny. Published in MBio, looks like it may be worth a mention. The researchers conducted a study that examined the nasal passages of healthy adults. 41% of those sampled had traces of triclosan in their nasal secretions, and the presence of triclosan in the secretions also correlated positively with nasal colonization by Staph aureus. When grown in the presence of triclosan, Staph aureus was found to be better able to attach to human proteins. Additional experiments found that rats exposed to triclosan were also more susceptible to nasal colonization by Staph aureus.
That was work from Blaze Bowles.
Yeah, do you know Blaze?
Sure, he's great.
Well, the great state, home of the Golden Gophers, has banned triclosan.
Triclosan, is that how you say it?
Minnesota has outlawed triclosan.
So this is saying that it makes things worse, basically, right?
Yeah. And so the state of Minnesota, probably using University of Michigan research, went ahead and banned the sale of triclosan.
All right. Thank you, Peter. That's pretty cool. Congratulations, Blue. And another cool article. And that'll do it for TWiM 79. You'll be able to find this at iTunes and also at microbeworld.org/TWiM. And do send us your questions and comments. You can email them to twim@twiv.tv. Michele Swanson is at the University of Michigan. Thank you, Michele.
My pleasure.
Good to have you back.
Thank you.
Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thank you, everyone.
And you can find me. I'm Vincent Racaniello, by the way, in case you didn't know that. You can find me at my blog, virology.ws. You can also find me on Twitter. My handle is PROFVRR. I want to thank ASM for their support of TWiM, Chris Condian in particular, and Ray Ortega for their technical help. Music that we use on TWiM, both at the beginning and end, is composed and performed by Ronald Jenkins. You can hear his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.